Hello students, welcome to EPG Pathsala. I am Professor Ramesh Kothari from Saurashtra University, Rajkot, Gujarat. Today we are going to discuss about a module on citric acid cycle as source of biosynthetic precursor under paper carbohydrate metabolism. So students, let's see what we are going to learn in this module. We will try to understand the function of a central metabolic pathway that produce several precursors. We will also try to see the importance of citric acid cycle intermediates in the synthesis of several amino acids and carbohydrates. We know that various amino acid synthesis begins at different points of citric acid cycle. We will also try to relate defect in biogenesis or anabolism required substrate formation of larger, more complex molecules. Roundabouts or traffic circles functions as hub to facilitate traffic flow. The citric acid cycle is the biochemical hub of the cell. Oxidizing carbon fuel, usually in the form of acetyl coenzyme A, as well as serving as a source of precursor for biosynthesis. The citric acid cycle is the central metabolic hub of the cell. It is the gateway to the aerobic metabolism of any molecules that can be transformed into an acetyl group or dicarboxylic acid. The cycle is also an important source of precursors, not only for the storage form of fuel but also for building blocks of many other molecules such as amino acids, nucleotide bases, cholesterol, and porphyrin, the organic compound of heme. Concept map of TCA as a source of biosynthetic precursor. This figure gives the overall of citric acid cycle, which is a source of biosynthetic precursor for many metabolic pathway. We synthesize carbohydrate, proteins, fatty acids, nucleotides, etc. from the intermediate of the citric acid cycle. Intermediate of the citric acid cycle. Intermediates of the citric acid cycle are drawn off as a precursor in many biosynthetic pathways, yielding the product in the shaded area. Shown in red are four anaplerotic reactions that replenish depleted intermediate of the citric acid cycle. The synthesis of all non-essential amino acids occurs through intermediates of major metabolic pathways like glycolysis, TCA, and pentose phosphate pathway. Based on their metabolic precursors, biosynthesis of amino acid can be grouped into 6 families. As shown in the table, amino acid biosynthetic families grouped by metabolic precursors. These several families like alpha-ketoglutarate, pyruvate, 3-phosphoglycerate, and oxaloacetate, phosphoenol pyruvate, and erythrose 4-phosphate, and ribulose 5-phosphate. Alpha-ketoglutarate gives rise to glutamate, glutamine, proline, and arginine. The three enzymes, namely glutamate dehydrogenase, glutamine synthetase, and aminotransferases, play a central position in amino acid biosynthesis by transforming ammonium ion into the alpha amino nitrogen of various amino acids. Figure shows the formation of amino acid glutamate, glutamine, proline, and arginine from alpha ketoglutarate. Glutamate and glutamine. Alpha ketoglutarate is converted into glutamate in a reaction catalyzed by glutamate dehydrogenase. In another amination reaction, glutamine is formed from glutamate in a reaction catalyzed by glutamine synthetase. Figure shows the glutamate dehydrogenase reactions in which the formation of water molecules and glutamate take place from alpha ketoglutarate. This figure shows the formation of L-glutamine from L-glutamate by the enzyme glutamate synthetase. The synthesis of proline crystallized derivatives of cyclic derivatives of glutamate is shown in figure. In the first reaction, an acyl phosphate is formed by phosphorylation of the omega carboxyl group of glutamate. It is further reduced by NADH to form glutamate gamma semialdehyde, the product which undergo cyclization and further reduction to form proline. Arginine is produced from glutamate via ornithine and the urea cycle. The ornithine biosynthetic pathway is similar to proline pathway but involve two additional steps to prevent cyclization by blocking the amino group of glutamate gamma semialdehyde as shown in figure. The acetylation of acyl acetyl coenzyme A block the alpha amino group of glutamate. This step would be followed transamination reaction in which the acetyl group is removed to yield ornithine. Most of the arginine formed in mammal is further degraded to form urea so that the available arginine is depleted. Thus, it is an essential amino acid and is growing animals eat indeed in more amount. Figure shows the biosynthesis of proline from glutamate. The figure shows biosynthesis of arginine from glutamate. Figure shows pyruvate as a precursor for biosynthesis of amino acids. 3 non-essential and 6 essential amino acids are synthesized from oxaloacetate and pyruvate. Figure shows oxaloacetate as precursor for synthesis of amino acids. Oxaloacetate is the precursor of asparagine, methionine, lysine, and threonine amino acid. The figure shows the asparagine synthetase reactions. Amidation of aspartate leads to the formation of asparagine. The NH4 is being donated by glutamine. The formation of lysine, methionine, and threonine occurs from aspartate. Aspartate beta semialdehyde deserve a branch point in an intermediate in all three pathways. Homocysteine serve as a precursor of methionine and threonine. The isoleucine and the valine pathway begin with the condensation of 2 carbon of pyruvate with either another mole of pyruvate, that is valine pathways, or with alpha ketobutyrate isoleucine pathways. The alpha ketobutyrate is derived from threonine in a reaction that require pyridoxal phosphate. The alpha ketobutyrate is derived from threonine, which play an important role in the metabolic reactions. Alpha ketoisovalerate, an intermediate in the valine pathway, is the starting point of fourth step branch pathways, which leads to the production of leucine as shown in figure. Figure shows the biosynthesis of leucine. Now, mechanism of conversion of acetyl coenzyme to fatty acid and steroid. Acetyl coenzyme A is a major biosynthetic precursor for the formation of lipids. Formation of fatty acids from formed molecule is called lipogenesis. Lipogenesis encompasses both the process of fatty acid synthesis and triglyceride synthesis, where fatty acids are esterified with glycerol to form fats. The products are secreted from the liver in the form of very low density lipoproteins and enters directly into blood, a site for their maturation, and delivers the endogenously derived lipid to peripheral tissue. Many of the enzymes for the fatty acid synthesis are organized into multi-enzyme complexes called fatty acid synthetase. The major site of fatty acid synthesis are adipose tissue and the liver. Given the number of biosynthetic products derived from citric acid cycle intermediates, this cycle clearly serves a critical role apart from its function in energy yielding metabolism. I would like to discuss history related with TCA cycle. The manuscript proposing the TCA cycle was submitted for publication to Nature but was rejected. It was subsequently published in Enzymologia. The Dr. Crabb proudly displayed the rejection letter throughout his career as encouragement for a young scientist. Pyruvate is converted to acetyl coenzymes before entering the citric acid cycle. The function of coenzyme A is a carrier of acetyl and other acyl group. So students, let us summarize what we have learned in this module. The citric acid cycle, which is also known as Krebs cycle or TCA cycle, mechanism of conversion of succinyl coenzyme A to porphyrins, where succinyl coenzyme A and glycine are combined by synthase to form sigma amino leucinic acid. Porphyrins are complex molecule containing a large heterocyclic organic ring structure in form of 4 modified pyrrole, that is 5-membered organic ring, subunits connected by methane bridge. Heme is a naturally occurring porphyrins in the biological system. It is comprised of ferrous ion complexed with 4 nitrogens of the specific porphyrin molecule identified as protoporphyrin 9 as shown in figure. Following figure shows the conversion of succinyl coenzyme A to porphyrin. If you see the route for the synthesis of porphyrin and heme, If you see the succinyl coenzyme A coupled with glycine to produce sigma amino levulinate, 2 sigma levulinate, amino levulinate condense to form porphobilinogens, and porphobilinogen in turn convert into 4 porphobilinogens. In second part of reaction, uroporphyrinogens 3 by the reaction of decarboxylation converted into coproporphyrinogen 3. Again decarboxylation reaction results into protoporphyrinogens 3, and this results in the formation of protoporphyrin 9, which lead to the synthesis of heme. All the intermediate of TCA cycle are potentially glucogenic, since they can give rise to oxaloacetate, and thus net production of glucose in the liver and kidney, the organ that carry out gluconeogenesis. If you see the mechanism of conversion of oxaloacetate to carbohydrate, gluconeogenesis is a pathway consisting of a series of 11 enzyme catalyzed reactions. The pathway may begin in the mitochondria or cytoplasm of the liver or kidney, this being dependent on the substrate being used. Many of the reactions are the reversible step found in glycolysis. Phosphophenol pyruvate carboxylase is a key enzyme in gluconeogenesis, which decarboxylates oxaloacetate to phosphoenol pyruvate, with GTP acting as the donor phosphate as shown in figure. Gluconeogenesis starts in the mitochondria with the formation of oxaloacetate by the carboxylation of pyruvate, and it is further reduced to malate using NADH, an essential step for its transportation out of the mitochondria. Malate is oxidized to oxaloacetate using NADH in the cytosol, where the remaining steps of gluconeogenesis take place. Oxaloacetate is decarboxylated and then phosphorylated to form phosphoenol pyruvate using the enzyme PEP. Molecules of GTP is hydrolyzed to GDP during this reaction. The next step in the reactions are the same as reversed glycolysis. However, fructose 1,6-bisphosphatase converts fructose to fructose 6-phosphate using one water molecule and releasing one phosphate. In glycolysis, it is phosphofructokinase 1 convert fructose 6-phosphate to fructose bisphosphate. This is also the rate limiting step of gluconeogenesis. Glucose 6-phosphate is formed from fructose 6-phosphate by phosphofructoisomerase. It is the reverse of step 2 in glycolysis. Glucose 6-phosphatase can be used in other metabolic pathways or dephosphorylated to free glucose, whereas free glucose can easily diffuse in and out of the cell. The phosphorylated form of glucose, that is glucose 6-phosphate, is locked in the cell, a mechanism by which intracellular glucose levels are controlled by the cell. The final reaction of gluconeogenesis, That is, the formation of glucose occurs in the lumen of endoplasmic reticulum, where glucose 6-phosphate is hydrolyzed by glucose 6-phosphatase to produce glucose and release an inorganic phosphate. Like 2 steps prior, this step is not a simple reversal of glycolysis, in which hexokinase catalyzes the conversion of glucose and ATP into glucose 6-phosphate and ADP. Glucose is settled into the cytoplasm by glucose transporters located in endoplasmic reticulum's membrane. The following pathways shows the conversion of oxaloacetate to carbohydrates. As shown in the figure, you can see during gluconeogenesis, glucose is converted to glucose 6-phosphate, which in part convert into fructose 1,6-phosphate. Now fructose 1,6-phosphate convert into fructose 1,6-bisphosphate. Enzyme is catalyzed by phosphofructokinase. This fructose 1,6-bisphosphate is converted into 3-phosphoglycerate, then 2-phosphoglycerate, and ultimately it finds its way into pyruvate. The TCA cycle take place in the mitochondrial matrix. And a final common pathway for oxidation of fuel molecule, which oxidize C2 unit. The enzymes pyruvate dehydrogenase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase are regulatory enzymes of the TCA cycle. The TCA cycle serves as a source of biosynthetic building blocks for the synthesis of nucleotide base, lipids, amino acids, and hemes. It is most likely that the citric acid cycle was assembled from pre-existing reaction pathways. As noted earlier, many of the intermediates formed in citric acid cycle are used in biosynthetic pathways to generate amino acids and porphyrins. Thus, compounds such as pyruvate, alpha ketoglutarate, and oxaloacetate were likely present early in evolution for biosynthetic precursors. The oxidative decarboxylation of 3 alpha keto acids is quite favorable thermodynamically. The elegant modulator structure of the pyruvate and alpha ketoglutarate dehydrogenase complexes reveals that how 3 reactions, that are decarboxylation, oxidation, and thioester formation, can be linked to harmless the free energy associated with decarboxylation to drive the synthesis of both acyl coenzyme derivatives and NADH. These reactions almost certainly form the core of processes that precede the citric acid cycle evolutionarily. Interestingly, alpha ketoglutarate can be directly converted into oxaloacetate by transamination of the respective amino acids by aspartate aminotransferases, another key enzyme of biosynthetic TCA cycle, which is shunting the synthesis of various amino acid family. Thus, cycles comprising smaller number of intermediates could have existed before the presence form evolved to harvest the electrons from pyruvate or other compound more efficiently. Is a nearly universal central metabolic, or I would say catabolic pathway, in which compounds derived from the breakdown of carbohydrates, fats, and proteins are oxidized to carbon dioxide, with most of the energy of oxidation temporarily held in the electron carriers, that is FADH2 and NADH. During anaerobic metabolism, these electrons are transferred to oxygen and the energy of electron flow is trapped as adenosine triphosphate. When intermediates are shunted from the citric acid cycle to other pathways, they are replenished by several anaplerotic reactions which produce 4 carbon intermediate by carboxylation of 3 carbon compounds. These reactions are catalyzed by pyruvate carboxylase, phosphoenol pyruvate carboxykinase, phosphoenol pyruvate carboxylase, and malic enzymes. Enzymes that catalyze carboxylation commonly employ biotin to activate carbon dioxide and to carry it to acceptors such as pyruvate or phosphoenol pyruvate. So students, this is all about citric acid cycle as source of biosynthetic precursors. Thank you.